Good day, this is Professor Will, CBMD PhD. Today is Sunday, February 21st, 2021. It is 12:12 and 53 seconds p.m. Eastern Standard Time. Today's research is on tuberculosis, so let's hop to it here. Tuberculosis. New biomarker indicates individual treatment duration. Date February 19, 2021. Source: German Center for Infection Research. Summary: the treatment of tuberculosis, TB, is long and demanding, in particular in cases of resistant tuberculosis. The WHO generally recommends a standard treatment duration of at least 18 months, as there are no reliable biomarkers for any early termination. Scientists have now succeeded in identifying a biomarker that points to an individual end of therapy based on the activity of 22 genes. In many cases, this probably allows the treatment to be shortened safely. When can tuberculosis therapy be stopped without risk of relapse? Doctors are faced with this question time and again. Because the lack of detection of the tuberculosis pathogen, Mycobacterium tuberculosis, is no guarantee for a permanent cure of the lung infection. Patients who respond to standard therapy may be out of treatment after 6 months, but for resistant cases, more than 18 months of treatment duration is currently advised. This is a very long time for those affected, who often have to take more than 4 antibiotics every day and suffer from side effects. Researchers urgently need a biomarker that enables the implementation of an individualized treatment duration. Researchers emphasize, after all, not every patient needs so long to recover. Since the absence of bacteria in the sputum does not justify a safe stop in therapy, the researchers set out to find alternative biomarkers in the patient. In collaboration with international tuberculosis centers, on the basis of patient cohorts, a model for the end of therapy could be developed that is based on an RNA determination in the blood. From many thousands of genes, 22 have been identified whose activity correlates with the course of the disease. The production of RNA of these 22 genes in human blood can tell us whether the patient is cured. It is an RNA signature from 22 genes, identified on 2 cohorts, and validated on another 3 cohorts. No other published transcription marker shows comparable properties so far. To identify this individual biomarker, the scientists within the DZIF have established 5 different patient cohorts. In all cases, these were adults who had contracted pulmonary TB, partly from non-resistant, partly from resistant forms. The individualization of the treatment duration is an important milestone on the road to precision medicine for tuberculosis. Even without progression values, one could risk to end a patient's treatment on the basis of this RNA determination. As a next step, the researchers are planning a prospective study at the DZIF. The aim is for patients in one study arm to receive treatment for as long as the biomarker suggests, while patients in the other arm receive treatment for as long as the National Tuberculosis Program recommends. The scientists then want to see whether the biomarker makes a shorter treatment duration possible. Hopefully, it will then be possible for patients with multi-drug resistant tuberculosis to save about 1/3 of treatment on average. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. I hope you enjoyed listening. Until next time, have a great day.